Thank you, Joel, for that nice introduction. I would like to thank our chair, Dr. Gelfand, for giving me the opportunity to be with you today and tell you a little bit about some of the work that we have done and others have done on this aspect. It's a pretty high bar to kind of cross to kind of talk about mechanistic data in this disease because there's still a lot of unknowns, but here we are. The title of my talk is Emerging Mechanistic Data Behind Psoriasis and Cardiometabolic Disease. These are my conflicts of interest. And just to kind of give you a very brief overview of the comorbidities of psoriasis. So it's very well recognized nowadays that psoriasis is a systemic inflammatory disorder, and it's astounding that about 75% of our patients have at least one or more comorbid condition, and this includes psychiatric, like we heard before here, pulmonary, hepatic, musculoskeletal, particularly psoriatic arthritis, renal, metabolic, malignancy, gastrointestinal, and then the cardiovascular aspects that I'm going to be talking to you about during the next 10 minutes or so. And this whole thing kind of was led by a landmark article published in the JAMA in 2006 by Dr. Gelfand and colleagues. And this was a very important study that really showed us that psoriasis is associated with cardiovascular risk. And this risk increase was particularly prominent among younger people with severe or more severe disease. This was followed up by a very impressive study by Nehal Mehta using PET imaging to kind of study inflammatory responses in psoriasis. So here you have a patient with fairly extensive plaque psoriasis. You can see that they're far on the left. And then you have the PET imaging of the body. And what you can appreciate, on the far left, you have a healthy control as a comparison. You can see the appearance of the plaques on the skin with the small arrows on the graphs, and you can see that those are lighting up, and that's reflective of the vascular inflammation, the increased metabolic activity that you can see in inflamed skin and psoriasis. But what's remarkable is how the entire vascular tree on the arterial side is kind of lighting up all the way from the heart all the way down to the small arteries in the extremities. And you can also appreciate that there's a low-grade increased uptake in the liver, kind of consistent with what we know is associated with steatohepatitis and psoriasis. This was kind of followed up by PET CT imaging, and again, a very striking and dramatic data. You can see there on the healthy control on the top on the left, and then you can see the same image in a patient with psoriasis where you have very marked increase in metabolic activity in the aortic arch. And remarkably, this increased activity actually correlates very strikingly with disease severity as measured by PASI. So the more higher the PASI is, the more severe the vascular inflammation is. And even when you correct for factors such as the sex or the age of the patient, it still remains very significant. And also if you correct for the Framingham risk factor scores, very significant and kind of linear correlation between disease severity and vascular inflammation and psoriasis. It's also been shown in kind of clinical trials or treatments. This is one that was published in Circulation in 2017. It was kind of following a group of patients with psoriasis, and then they followed the group for over a year when they were treated, and then looked at what happened in the patient that actually responded to treatment. So as you would expect, the disease severity got better as measured by PASI, but multiple other things actually got better along the way, and that's in this kind of a group over here. I know it's hard to kind of see individual data in this table, but what they saw was actually improved blood pressure in these individuals. There was decreased systemic inflammation. There was also a decrease in the coronary plaque burden. So again, very convincing correlation and association between psoriasis and cardiovascular disease, and also changes with effective treatment in psoriasis. But these are associations, and again, one way to kind of try to address, causality, what direction is the effect, is this fairly recent tool called Mendelian randomization tool. So what that does, it uses genetic variation as a natural experiment to investigate the causal relation between potentially modifiable risk factors and health outcomes in observational data. So it is in between, case control or association studies where you have strong association, but weak evidence of causation, and then these interventional kind of trials, we have strong evidence of causation. So these Mendelian randomization trials kind of are in between the observational studies and then the proof of concept, the interventional trials. So what we did, what our group at Michigan did, is we kind of looked at this using some of the genetic data that we have access to. And this includes 70,000 cases and about 200,000 healthy controls, combining both psoriasis and patients with atherosclerotic disease. So what we found is that when we looked at the, you have psoriasis there on the left, and you have the cardiovascular disease on the left. So when we look at the cardiovascular risk factors, we actually found that the cardiovascular-associated genes actually associated more strongly with psoriasis than psoriasis with cardiovascular disease. And we can also reproduce many of the same risk factors for cardiovascular disease, such as BMI, waist-hip ratio, and type 2 diabetes, and high cholesterol. So what does that mean? It kind of indicates that this is a bit surprising. We did not expect this when we saw this data, but it indicates that the direction of the effect is maybe more from the cardiovascular disease on the skin than actually from the skin to the cardiovascular disease. So kind of turning things a little bit on their head. We can see the effect a little bit. There's a weak effect from the skin to the atherosclerotic plaque, but the predominant and most of the effect actually is in the other way around. And if you look at it in terms of the gene correlation, you can see that the grouping of the diseases that are kind of going together is the type 2 diabetes, the body mass index, the cardiovascular disease, high cholesterol. We also see asthma kind of linking in with those two, but psoriasis kind of remains outside with much weaker association with the cardiovascular disease. However, the genetics can be useful, and again, you can use that as a way to kind of identify the shared mechanism. So this is kind of using and analyzing the shared and distinct genes that have an overall association score of about 0.1. And then we're looking at which ones are shared between psoriasis and which ones are unique to each condition, whether it's psoriasis or cardiovascular disease. And what you can see here is that many of the targets that we have kind of been focusing on in psoriasis over the past 2 decades, such as TNF and IL-17A, are shared between psoriasis and cardiovascular disease. And this is perhaps not surprising. If you look at the immunology of cardiovascular disease, and particularly the T cell cytokines that have been implicated in the pathogenesis, many of these are also found in psoriasis, and that includes the Th1, Th17, and even T regulatory cell function. And if you look at kind of mouse experiments that have been done, many of these have shown that IL-17A itself is increased in atherosclerosis. And if you block IL-17A, you actually manage to decrease the size of the atherosclerotic plaques in mice. And there's also additional work published in Science Translational Medicine a few years back showing that the mechanism actually is quite complex, with interaction between different inflammatory mediators, such as TGF-beta, kind of stabilizing the atherosclerotic plaque in an IL-17A dependent manner. So complex interactions. If we look at the ones that are kind of shared at a genome-wide significance level between psoriasis and atherosclerotic disease or cardiovascular disease, we have found 3 different molecules. One of them is this TEX1, which is a linear non-coding RNA. But we also find IFIH1, which encodes for RIG-I, and also the IL-23A, which encodes for the P19 subunit of interleukin-23, as being kind of shared between psoriasis, which is the top panel, the cardiovascular disease shown in the middle, and then the trans-disease meta-analysis that you can see on the bottom panel. There's not much known about TEX41 in either psoriasis or cardiovascular disease, but we know a lot about IL-23. It's been a major target in psoriasis for, I think, well over a decade now. It has a key role in T cell function in terms of driving and stabilizing a Th17 phenotype. And then it also has a role in inflammatory bowel disease, particularly Crohn's disease, through an effect likely on macrophage dendritic cells and kind of driving pro-inflammatory responses there. And that's one of the reasons why the anti-IL-19 drugs actually work fairly well for IBD. The involvement of this cytokine in atherosclerosis is still not well understood, though. IFIH1, RIG-I, this is a sensor for double-stranded RNA viruses and is a cytosolic sensor. It activates inflammatory responses, particularly interferon responses, and by that kind of drives an autoimmune or inflammatory kind of a phenotype. But it's interesting when you kind of look at the ones that are clearly shared between psoriasis and cardiovascular disease, is that the commonality between the two actually is inflammatory response. So what about mechanisms? What has been done in this field? There actually is quite a bit of work showing that skin actually does and may amplify and accelerate atherosclerotic process. One of them is a model that was published now over 12 years ago by my colleague Nicole Ward. She's a mouse expert and has generated the majority of the mouse models that we have looked at in terms of psoriasis susceptibility. And this is the Tie2 model that is overexpressed in keratinocytes and drives this kind of inflammatory phenotype that you can see there on the left with this red scaly skin that the mice have. And if they cross this mouse with an ApoE deficient background, and then you give them high fat or atherogenic food, you can actually induce atherosclerotic plaque in these mice. These plaques are a little bit different than what you can see in humans. They're not in the coronary arteries themselves. They're actually in the aortic root right next to the cusps of the aorta, like you can see there. And it usually does not involve the coronary arteries. And this actually happens fairly quickly in these mice. In a matter of a few weeks, you actually get fairly well-developed plaque. And you can see there that compared to the control in the middle panel, that the KC Tie2 model actually has fairly marked inflammatory infiltrate. You can see the trichrome staining with the disruption on fibrosis in the plaque, and then disruption of the internal elastic lamina on the bottom. And it's quite remarkable that in this model, when you turn the inflammatory phenotype off with doxycycline, you can actually reverse some of that inflammation in the aorta. So again, providing evidence that you may be able to kind of offset it. There is a link between the two. Another more recently published study, this was published in JCI just a few weeks ago. This is looking at the cathelicidin, or LL37, which is a pro-inflammatory antimicrobial protein, highly expressed in psoriasis, but also expressed in cells such as neutrophils and other immune cells. But when they make a transgenic mouse and put them on this ApoE deficient model and the atherogenic diet, they can actually see that the cathelicidin mouse, the LL37 mouse, had accelerated lipid uptake in the aorta. You can see that on the oil O red stain in B, and they have increased plaque area of the atherosclerotic plaque. Fairly dramatic phenotype and independent of the total cholesterol and the LDL. And in this model, what the authors showed in a fairly elegant way is that LL37 binds to LDL particles in the blood, will facilitate its uptake by macrophages, and you will get these foamy macrophages that then drive atherosclerosis. And again, showing a way whereby skin inflammation through a specific mediator may actually affect and accelerate atherosclerotic process. What about the other way around in terms of the vascular inflammation kind of driving skin inflammation? The data here is much more sparse, but there's one study published last year that actually I thought was quite interesting, and that is focusing on vascular inflammation, particularly the glycocalyx, which is on the inside of these cells. I don't know if you can see this. It kind of shows you the extracellular component organization, and particularly the glycosaminoglycans. You can appreciate the glycocalyx here in healthy capillaries here in the skin, and then you can appreciate how the glycocalyx actually is very disrupted in diseases such as psoriasis. Markedly diminished in both in terms of the thickness and the coverage. And you can appreciate that here by validation with the hyaluronic acid that actually will stain the glycocalyx. You can see that here in the healthy vascular structure, but again, completely absent in psoriasis. So you have a situation like is shown here in this kind of schematic, where in healthy endothelium you have a very healthy and robust glycocalyx that actually regulates efflux of immune cells into the tissue. With inflammation, you have a breakdown in that glycocalyx. You get a decrease in the thickness and the overall coverage, and that leads to increased inflammatory infiltration in the skin. And this is one mechanism whereby vascular inflammation may actually help to kind of drive or amplify inflammatory responses in the tissue. So in a way, we have still a lot of work to kind of do. You know, our view of cardiovascular disease and psoriasis has been for like a long time, we treat the skin, and when we treat the skin, the inflammation goes down, and with that, the systemic inflammation decreases, and thereby we kind of diminish or decrease the risk of cardiovascular disease. But I think it may be more complicated. It might be a bidirectional kind of an effect, and the treatment that we are using to treat psoriasis may also be directly affecting cardiovascular disease. And with that, we might be decreasing systemic inflammation by targeting both of these organs. So just to quickly summarize, I know I'm a little bit over time. So multiple comorbidities associated with psoriasis, including cardiovascular disease. The causal link may not be as straightforward as we have believed. The confounding issues, other psoriasis-associated comorbidities may have a direct causal link to atherosclerosis, such as type 2 diabetes, body mass index, and cholesterol and triglycerides. The direction of the effect appears to be a bit stronger from the cardiovascular risk to psoriasis than vice versa. But again, there's evidence of bidirectional effect, particularly both to some extent from the Mendelian randomization studies, but also from these various animal mouse models that I've showed you before. And it seems to be kind of linking through that inflammatory process on both sides. The mechanisms involved are still very unclear, and it's actually quite difficult to study these, given the shared and overlapping immune pathways between these two conditions. But I would like to stress, that this does not really take away the necessity of informing patients about cardiovascular risk. These two conditions are strongly associated, and the involvement of dermatologists in screening for these diseases is of high importance. So thank you for listening, and I'm happy to take questions.
We'll take questions.